welcome to this explainer. We hear about these massive medical breakthroughs all the time, right? Something performs absolute miracles in a petri dish and then crickets. Why does it take a decade, millions of dollars, and just a completely insane gauntlet of regulations to actually reach a human patient? Well, today, we're going to break down that staggering complexity. We're pulling back the curtain on the hidden realities of bringing a cutting-edge therapy, specifically extracellular vesicles, from a tiny lab to full-on clinical-scale manufacturing. Let's get into it. So here's our roadmap for today. First, we'll look at the clinical miracle, then the lab to commercial chasm, next, the 5-day engine, the QC gauntlet, intellectual property hurdles, and finally, we'll wrap up with the biotech endgame. All right, starting with section 1, the clinical miracle. This is the massive patient win that kicks off this whole crazy journey. Okay, so our focus here is on producing extracellular vesicles, or EVs, and these are derived from cardiac progenitor cells. Think of these CPCs as basically the biological heroes of our story. They're specialized stem cells derived from induced pluripotent stem cells, and they secrete these tiny, powerful little packages, the EVs. And these EVs can actually help repair damaged tissue. But, the challenge isn't just discovering them. It's figuring out how to engineer them at a massive clinical scale. And here's why people are trying so hard to scale this up. The real-world results are they're profound. Look at this phase 1 clinical trial for severe heart failure patients, specifically NYHA class 3. We're talking about people who are literally gasping for breath after walking just 10 meters. But after getting targeted EV doses, and I mean massive doses, like up to 40 billion EVs, they progressed to class 2. They went from struggling at 10 meters to walking 100 meters easily. It's a complete game changer for their quality of life. Which brings us to section 2, the lab to commercial chasm. This is where we have to somehow bridge the gap. To really understand this chasm, we have to look at two completely different worlds. On one side, you've got RUO, or research use only. This is the lab. It's flexible, it's all about discovery, and it totally tolerates a bit of biological variability because, hey, you're just proving a concept. But on the flip side, you have CGMP, current good manufacturing practice. This world is unforgiving. It mandates absolute sterility, intense risk mitigation, and perfect, unyielding consistency. Seriously, what works perfectly fine in RUO will literally get you shut down in CGMP. It's basically a massive structural gap. Think of the research phase like a simple wooden pallet. It's good enough to hold a few boxes of, early discovery. But CGMP manufacturing, that is a highly engineered, heavy-duty steel scaffold. It is built from the ground up to withstand brutal inspections, ensure absolute sterility, and guarantee that the 1 billionth EV you produce is completely identical to the very first one. So making that jump to CGMP requires some really massive structural upgrades. You're not just moving from a normal lab bench to a slightly cleaner bench. No, you are moving into a highly controlled grade B cleanroom that houses an even stricter grade A processing zone. Your casual lab notebooks? Gone. Replaced by strict, standardized risk assessment frameworks for every single tiny step. Even your raw materials need insane documentation now. Certificates of analysis, certificates of origin, rigorous safety approvals, the whole shebang. All right, section 3, the 5-day engine. Let's look at this meticulous manufacturing process. The mechanics here basically run in 3 core steps. Step 1 is upstream cultivation, that's thawing and growing your cells. Step 2 is a critical high-stress transition called the palm swap. And step 3 is downstream isolation, where you harvest and purify the final product. Sounds pretty straightforward, right? But on a microscopic level, it's incredibly complex. Here's how it flows over 5 days. Days 1 to 3, you're really just stabilizing and cultivating those cardiac progenitor cells. But day 3 is the inflection point. The cells get swapped into a production medium, or POM. This swap actually stresses the cells out on purpose. And as a survival response to that stress, they release the secretome, the EVs we want, into the medium over days 4 and 5. After that, the whole thing goes through intense inline filtration and tangential flow filtration to isolate and concentrate that final, precious product. Moving on to section 4, the quality control gauntlet. And this requires a totally bifurcated architecture. So because producing EVs is this dynamic, living, biological process, quality control can't just check the final product at the end and call it a day. It requires this bifurcated approach. You have to simultaneously monitor the engine, meaning the living CPC cells that are actually producing the EVs, and the output, the final extracellular vesicles themselves. Because think about it, if the engine breaks down or mutates, the output is completely useless. Let's look at the engine first. We have to make sure these cells don't differentiate improperly when we stress them out with that day 3 medium swap. Remember, these are progenitor cells. Their natural instinct is to turn into other things. So manufacturers have to continuously track identity markers like CD56 and CXCX4 using flow cytometry. They also have to run genetic stability assessments like karyotyping just to be absolutely sure the chromosomes haven't mutated under all that manufacturing stress. And this, right here, is where the money absolutely evaporates. The hidden costs are staggering. Keeping up with these strict cell identity markers is not cheap. You're running continuous sterility screens, complex marker analysis, and on top of that, you have mandatory 3-year stability testing protocols. That means operating specialized negative 65 degrees Celsius freezers for years. You are literally paying teams of highly trained scientists just to monitor ice age level freezers for 3 years straight. It's wild. Then, moving from the engine to the output, that final Secretome product has to be rigorously verified. You've got physical specifications to meet, mapping the exact particle size distribution. You've got safety purity screens checking for any leftover host cell protein residue or endotoxins. And crucially, you have to prove its biological identity. You need to confirm the presence of essential exosome markers like CD63 and CD81, because if those aren't there, you don't have medicine. You just have a very, very expensive vial of biological soup. And finally, before any of this even comes close to a human patient, the functionality has to be definitively proven. It's not enough that they just look like EVs, they have to act like them in the real world. So they do strict proliferation assays using umbilical vein endothelial cells, angiogenesis scratch tests to see how fast cellular repair happens, and mandatory in vivo toxicology profiling just to guarantee you aren't accidentally triggering tumors. Every single batch has to survive this gauntlet. Okay, Section 5, Intellectual Property Hurdles. Let's talk about the big commercial bottlenecks. So you've survived the manufacturing gauntlet. Great, but navigating the IP bottleneck is a whole other nightmare. It's this tangled web of foundational constraints. For instance, just using iPSCs to even start your process, heavily locked down by existing patents. Then you have process constraints, overlapping IP rights that cover specific isolation methods or filtration techniques. You could literally successfully develop a life-saving therapy, only to find out there is no way you can legally sell it without signing these crippling sublicensing agreements with like 5 different companies. Which brings us to our final section. Section 6, the biotech endgame and the harsh macroeconomic reality. Let's put this cash burn into perspective. Think about a traditional hardware tech model. A giant like Asus can generate roughly 60 billion new Taiwan dollars a month. They release a product, boom, immediate consumer revenue and feedback. Biotech is the exact opposite. These companies are burning millions of dollars daily, running those stability tests, jumping through clinical hoops, and they will see absolutely zero consumer revenue for over a decade. It is just an extreme macroeconomic contrast. On one side, you have the high-volume, rapid iteration tech model, and on the other, this massive, decade-long cash burn required to treat maybe just one specific patient cohort in a clinical trial. You're funding grade B clean rooms and highly specialized scientific talent for years before a single dollar of actual profit comes back. It requires incredibly deep pockets and, honestly, an iron stomach for risk. So because of that insane cash burn, the true exit strategy for these life science startups is almost never to build a massive standalone pharmaceutical sales empire from scratch. No. Step 1 is absorb the risk. Step 2, prove the science works in phase 1 and 2 trials. Step 3, acquisition. The entire goal is to de-risk the startup just enough so that big pharma swoops in and buys the company. They acquire the IP, that perfect CMP process, and the clinical data, and they just plug it into their own massive global pipelines. Which leaves us with this final kind of provocative thought. We spend all our time thinking about the patient, right? The person gasping for breath at the 10-meter mark. But in the actual business reality of this clinical manufacturing gauntlet, the ultimate customer for a medical breakthrough startup is actually big pharma. So how should that reshape our understanding of medical innovation, and who is it truly designed to serve first? Definitely something to think about as you keep exploring the incredible, hidden mechanics of the world around us. Thanks for joining me on this explainer.